Hi, my name is David Glubzinski, and I'm an undergraduate student at the University of Michigan studying environmental engineering. This summer, I've had the opportunity to work with, under the direction of Professor Nancy Love, Dr. Kelly Martin, and graduate student Lauren Stadler on two projects involving sustainable wastewater treatment. The first project involves sustainable nutrient removal downstream of an anaerobic membrane bioreactor. The second project involves determining the impact of dissolved oxygen on pharmaceutical removal within a wastewater treatment process. Let me show you a little bit about what these two projects are all about. Before I introduce you to the reactors, let me give you a bit of background information on the downstream system. In most wastewater treatment plants today, aerobic bacteria, or bacteria that require oxygen, are used to remove organic matter and nutrients from the water. However, this process accounts for between 40 and 60% of the energy demands of the entire treatment process. To reduce energy requirements, research on anaerobic membrane bioreactors, or AnMBRs, is being conducted by another group in our lab. These systems use bacteria that do not require oxygen, which leads to major energy savings from not having to constantly aerate the water. Additionally, these bacteria also produce methane as a natural byproduct, which can be used to generate energy. However, the bacteria in AnMBRs do not remove nutrients such as nitrogen-containing compounds from the wastewater. If untreated, these nutrients can ultimately lead to algae blooms, fish kills, and other harmful ecological impacts on our environment. That is where our downstream system comes into play. In a traditional nitrogen removal process, ammonia is first converted to nitrite and then to nitrate by aerobic bacteria before the anaerobic bacteria convert the nitrate to nitrogen gas. To improve the sustainability of nitrogen removal, our downstream system uses both ammonia oxidizing bacteria, or AOBs, and anammox bacteria. The ammonia is first converted to nitrite by AOBs, and then the anammox remove the nitrogen in the form of nitrite from the water by converting it to nitrogen gas. This process saves 25% of the energy demands and 40% of the carbon demands of conventional biological nitrogen removal processes. This means that less carbon is needed for the nutrient removal reactions, and therefore more carbon can be converted to methane by the AnMBR. Additionally, dissolved methane that reaches the downstream system is removed by methane oxidizing bacteria, therefore minimizing potential contributions to greenhouse gas emissions after discharge into local waterways. Now that all the details are out of the way, let's take a look at the reactors. For the downstream system, we built two different types of reactors. First is the membrane biofilm reactor, as shown here. This reactor, if you look closely, you can see individual fibers running up the side of the reactor. These are the membranes on which the biofilm grow. How it works is that air travels slowly up through the center of the hollow fiber membrane, and as it travels up, it slowly diffuses through the membrane wall and is delivered to the biomass growing on the membrane. This configuration allows oxygen to be passively delivered to the biofilm. Shown here is a cross-section of a hollow fiber membrane. In blue is the membrane wall. The AOBs grow right on the outer surface of the membrane, closest to the source of oxygen. This creates favorable growth conditions for the anammox, because the AOBs consume all of the oxygen present. These two bacteria are syntrophs, because the AOBs produce the nitrite that the anammox need to generate energy. Up here we have a series of probes. The first probe measures nitrate/ammonia concentrations. The second probe measures the pH of the system. And the third probe measures the oxidation reduction potential, or ORP, of the system. Moving down here, this is where the influent to the system is stored. This influent is a synthetic anaerobic effluent, which we make every couple of days. The second downstream reactor is the sequencing batch reactor, as shown here. This reactor is made up of tiny, spherical aggregates comprised of both AOBs and anammox. This reactor operates like a batch reactor, with two stages of treatment in each cycle. First is the aerobic stage, in which oxygen is vigorously bubbled up from the bottom of the reactor. This is the condition in which ammonia-oxidizing bacteria thrive. Second is the anaerobic stage, in which there is no dissolved oxygen present. The anaerobic stage provides favorable growth conditions for the anammox. Like for the MBFR, a synthetic anaerobic effluent is fed up from the bottom tank into the SBR. Up here we have a series of probes which measure the pH, the dissolved oxygen, and the nitrate/ammonia concentrations in the system. My main responsibility for these two reactors were to both sample and test reactor influent and effluent for ammonia, nitrite, and nitrate. These parameters help us to evaluate how each reactor is performing. But that is not all. This summer, I've also been working on determining the impact of dissolved oxygen on pharmaceutical removal within wastewater treatment. When you take a pill, your body only absorbs a fraction of the actual dosage, and the rest goes out the other end and ends up in the wastewater treatment plant. If pharmaceuticals are not degraded within the wastewater treatment plant, then they will move on to local water bodies and affect the ecology of our rivers and streams. Let's check out the setup of the pharmaceutical project. Here we have a series of 3 reactors. The first reactor is under high DO conditions, and the second and third reactors are under low DO conditions. The bacteria in each of these reactors are fed with a synthetic influent, which I make every couple of days. Through running many long batch experiments, and frequent testing of ammonia, nitrite, total suspended solids, and nitrate. We have been able to determine that low oxygen microbiome communities are more diverse and are better at degrading certain pharmaceuticals such as ibuprofen and sulfamethoxazole. Overall, my research experience in Michigan this summer has been top-notch. I've definitely learned a lot, and I've really enjoyed working with all of my awesome teammates in the Love Research Group. Thanks for watching, and as always, go Blue!